My name is Dr. Patel, Professor and Chief Soil Health Specialist, Anand Agriculture University and Parul University, and Mati Agro Mart Private Limited, Anand, Gujarat, India. So today, the topic of my YouTube is role of micronutrients, boron, molybdenum, chlorine, cobalt, and nickel in crop production. The importance of micronutrient agriculture production. Micronutrients are required by the plant in lesser quantities, yet essential for the plant growth and survival. Micronutrients are essential elements that are used by plant in small quantity. Yield and quality of agriculture products increase with micronutrient application. Therefore, human and animal health is protected with feed of enrichment plant material. Each essential element only when can perform its role in plant nutrition properly that other necessary elements are available in balance ratio for plant. So when we will apply the balanced nutrition in soil, then plant will take the balanced plant nutrition and better growth of the plant, and we are getting the higher crop production. So how we can know the initial nutrient of status? So for that we should have to carry out soil analysis before sowing or planting of the crops. It is absorbed by plant under undissociated boric acid. So first we have talked about the boron. So boron and molybdenum are immobile in plants, so deficiency symptoms appear in young leaves because both are immobile in plant. Boron deficiency found in sandy soil, organic soil, and fine texture soil because it is laid down in a sandy soil because sandy soil is highly permeable. Then function of boron, new cell development in meristematic tissue, pollination, root, seed set, translocation of sugar, starch, nitrogen and phosphorus, synthesis of protein and amino acid, nodule formations in legume, regulation of carbohydrate metabolism. Then deficiency of boron. Since it is immobile, deficiency symptoms occur in terminal bud growth or new growth of the plant or top of the plant because it is a boron is a immobile in the plant. Flowering and fruit development are restricted. Sterility and malformation of reproductive organs, thickened and cull leaves, discoloration, cracking or rotting of fruit, tuber or roots, apple internal cracking, breakdown of internal tissue in root crops, given rise to darkened areas, referred to brown heart, black heart, cotton whipping disease. So it is due to deficiency of the boron. So boron deficiency in corn, maize. Here you can see boron deficiency. Here extreme boron deficiency. So you can see the leaf scratches are found and yellowing of the leaf. Then corn cob also affected. Then boron deficiency in lucerne alfalfa. So you can see that when the boron deficiency in the soil, then the growth of the lucerne crop is poor, and you can see the chlorotic on the top leaves. Then boron deficiency in the cotton, so cotton really decline 15 to 20%. Then boron deficiency in potato. Then boron deficiency in cauliflower. It is also known as whiptail. So we can see in further. Next slide. Then boron deficiency in cowpea. You can see here typical deficiency of the boron in cowpea. Then boron deficiency in tomato crop. Then boron deficiency in mango. You can see the fruit cracking of the mango fruits. Then boron deficiency in banana crop. So you can see the stunted growth and yellowing on the top leaves. Then boron toxicity in rice. You know that when the boron is greater in a soil, higher in soil, then there is a toxicity in the crop. So you can see here rice, boron toxicity inhibit the formation of starch for sugar and affect the formation of B carbohydrate complexes. Boron toxicity in citrus. that is lime crops. You can see here margin are chlorotic and yellowing of the leaf. So it is a boron toxicity. So boron toxicity is also harmful. So we should have to supply the balanced nutrition of the boron. And how we can know? So we can know only through the soil analysis before sowing or planting of the crops. Then again you can see that molybdenum is also important. So molybdenum deficiency in alpha leucination. So it is a deficient plant and it is sufficient plant with the molybdenum. So molybdenum deficiency in tomato plant you can see here. So molybdenum deficiency prevents the fixation of nitrogen. So it related with the nitrogen fixation. So molybdenum is required for the better nitrogen fixation. This process involves higher plant and symbiotic organisms such as rhizobium, and so molybdenum deficiency can produce symptoms associated with the deficiency of nitrogen. So when there is a molybdenum deficiency in soil, then nitrogen fixation prevents, and we can also observe the deficiency of the nitrogen because nitrogen is not uh, get sufficient by the plant, so you get the deficiency of the nitrogen. And you can know that the deficiency of the nitrogen you will find on the lower leaf of the plant. If the molybdenum is needed directly by the plant for nitrate reductase, then symptoms peculiar to molybdenum occur, although essentially the plant can be considered to be suffering from shortage of protein due to failure of the initial process of reduction. So you know that nitrogen is less available to the plant and there is a shortage of protein. So protein synthesis is lesser when there is deficiency of the molybdenum. So in alkaline soil, molybdenum becomes more soluble and it is accessible to plant mainly in its anion form as MO4. In contrast, in acidic soil, pH lesser than 5.5, molybdenum availability decreases as anion absorption to soil oxide increases. So we should have to remember that molybdenum is a exception micronutrient that is more available in alkaline soil, whereas it is less available in the acidic soil. Then molybdenum, non-metal anion absorbed by molybdenum. It is weak acid and form complex polyanions such as phosphomidate. Plant contain less than 1 ppm molybdenum. So function of molybdenum, essential component of enzyme nitrate reductase, we have seen, we catalyze nitrate to nitrite. Structure component of nitrogenous enzymes involved in nitrogen fixations, we have seen, essential for absorption and translocation of ion in plant. Then deficiency of molybdenum, so we have discussed molybdenum deficiency in cauliflower, causes a condition called whiptail, in which the leaves have a thin, stripy appearance. So inhibit flower formation, imbalance various amino acid in plant, reduce activity of symbiotic and non-symbiotic nitrogen fixation. So cauliflower typical deficiency that is called whiptail. Then next, micronutrient chlorine. Normal concentration in plant is about 0.2 to 2% absorbed by plant as chlorine through roots and aerial parts. Function essential for biochemical reaction, osmotic cations, neutralization. Reactors act as a counter ion during rapid potassium fluxes involved in the evolution of oxygen in photosynthesis, create disease resistance by increased osmotic pressure in cell sap. So deficiency of chlorine, partial wilting and loss of turgidity, necrosis, lip bruising, and reduction in growth. So here you can see that chlorine deficiency in durum weight. Here chlorine sufficient plant. Here chlorine deficient plant. So here boron is sufficient. chlorine is sufficient. So when chlorine is sufficient, then the healthy plant. And when the deficiency of the chlorine in during wheat, so you can see the clear cut differences with chlorine and deficiency of the chlorine. Then chlorine deficiency in wheat crop. So in left side, you can see the deficiency of the chlorine. On right side, you can see the sufficiency of the chlorine. So healthy growth. Here also called chlorine deficiency in tomato crop. You can see the yellow ring. So chloritic leaf production of the tomato has been declined. Then chlorine toxicity. So when there is a chlorine toxicity, then there will be a scorching of the leaf. So you can see the crotching of the leaves. Trees with scorched leaves have brown or dead tissue on the tips. So you can see the dead tissue of the tips. Margin you can also see, or between the vein of the leaf. Or you can see here the between the vein of the leaf. Leaf may be smaller than usual. So here the healthy plant with the chlorine, here the toxicity. So toxicity is also harmful. And therefore balanced nutrition should be applied. And therefore we should have to carry out a soil analysis before sowing or planting of the crop. And you can see the leaves may be yellow and drop. Finally, eventually, the leaf may be dropped and it is a healthy plant. Then cobalt, normal concentration of cobalt in dry matter 0.002 to 0.5 ppm. Function of cobalt, essential for fixing atmosphere. Nitrogen, it forms vitamin B12 during growth and development of symbiotic, improve growth, transpiration and photosynthesis, take part in leg hemoglobin metabolism, and ribonucleotide reductase deficiency, acidic deficiency, the acidic, highly rich sandy soil with total cobalt low. So it will be found deficient in acidic soil or highly rich soil. Highly rich soil is sandy soil. So you can found the deficiency of the cobalt in sandy soil or highly rich soil. So most forages are good source of cobalt with a range of 0.1 to 0.3 ppm diameter or milligram per kg or ppm. Overly drained soil tend to lead to higher concentration of cobalt in herbage grown on this soil. Overliming soil to increase the pH above 6 will reduce the ability of the cobalt and may lead to deficiency. So when we are applying the overliming, then it increases the pH above 6 and it will reduce the ability of the cobalt and may lead to deficiency. So overliming also not beneficial. So we should know by the soil analysis. So cobalt deficiency. Then function of silica. So contribute structure of cell wall, contribute drought tolerant to crop, regulate photosynthesis. Then factor affecting silica availability. Higher water content encourages silica uptake. Heavy application of nitrogen decrease silicon concentration. So heavy application of nitrogen decrease silicon concentration in the plant. Liming decreases silicon uptake in plant. So we have seen. Then acidification increases silica uptake. So in acid soil, silica is more increases. So more uptake of the silica. Iron and aluminum oxide influence uptake of silica by plant. So in acid soil, you can know that there will be dominance of the iron and aluminum. So in acid soil, aluminum and iron influence the uptake of the silica by plant, particularly acid soil. And in India, we are observed the acid soil in Assam, Himachal Pradesh. Then silica fertilizers, calcium silicate slag, 18.21% silica, calcium silicate, 31% silicate, sodium metasilicate, 23% silicate. So you can see here the soft droppy leaves and comes of rice plant. Leaves become chlorotic. So you can see the leaves become chlorotic will later become necrotic brown spot. So you can see here the necrotic brown spot. Entire leaf become brown or pink. So color is brown and pink color. Reduced panicle per square meter, susceptible to lodging. So when there is a deficiency of the silica, then plant will be loss of the rice, and we are losing the rice crop production. So correction measure, apply calcium silicate at the rate of 120 to 200 kg per hectare, potassium silicate at the rate of 40 to 60 kg per hectare for disappearance of the same plant. So you should have to remember that rice and sugarcane crop require higher silica as compared to the other crops. And both these crops are highly responsive when the deficiency of the silica in a soil. So effect of silicon on bioproduce oryza, this is rice. So either silica leaf, jack and silica soil. So you can see here the better growth. Silica foliar content percentage. So you can see here. Then last one is nickel. Nickel content in plant is 0.1 to 1 ppm dry matter basis taken by plant as nickel plus 2. High level of nickel may induce zinc and iron deficiency. So nickel have antagonist effect with the zinc and iron deficiency. So when there is a high level of the nickel, then there will be deficiency of zinc and iron. Then function, nickel metal component of ureas that catalyze reaction, cobalt, ammonium plus hydrogen, plus water, so is equal to ammonia and plus carbon dioxide. Essential for nitrogen metabolism, stimulate nodule weight and yield of crop. So nickel is also important. So effect of nickel treatment on growth of wet root after 1 days, after 4 days, and after 7 days of exposure to the metal. So you can see here the growth of the wet roots after 1, 4, and 7 days. So you can see here clear. A 1 days, B 4 days, and C 7 days exposure of the metal. So you can see here the better growth. Then hidden hunger bougainvillea. So it is a ornamental plants. So nickel can be potentially green up foliage. So it is also required. So hidden hunger means we cannot see with the naked eyes, but there is a deficiency of the nickel in the plant. And how we can know? For that we should have to carry out the plant analysis. So hidden hunger appears to be fairly common in certain ornamental species growing in artificial soil media. So it is a nickel deficiency. So you can you can remember that when there is a hidden hunger, then the crop production has been declined about 10%. So by knowing the plant analysis, we can know that which deficiency, which hidden hunger in the plant. So different types of hidden hunger of the micronutrient, zinc, iron, nickel, and so on. The nickel deficiency in cannabis, marijuana, drugs. So it is a drug plant. So you can see that it is a deficient plant. It is a healthy plant with the nickel. So nickel deficiency, lab grow. Whereas in right side, you can see the nickel sufficient. So it is also in the cannabis. So it is a cannabis, marijuana, drug plants, and people are taking. Then nickel toxicity in maize seedling. So you can see here toxicity is also harmful, and therefore we should have to carry out the soil analysis, and as per the diagnosis of the nutrients, or knowing the initial nutrient status, we should have to apply the particular nutrient in a soil. Suppose you have not carried out the soil analysis of the nickel, and suppose your soil have a nickel toxicity or high, then you can see the growth of the maize seedling has been declined. So here you can see the control plot, here nickel. So when there is a higher growth, then crop has been. So nickel toxicity in base. So nickel toxicity here, the nickel is supposed in a high in soil, and you also apply the nickel in soil, then there will be toxicity. And naturally also sometimes nickel is present in higher concentration. So here you can see the when the toxicity, then you can see the tip of the maize seedling is chlorotic, and margin also become chlorotic. So if you like my YouTube on micronutrients in agriculture, function, deficiency on the plants, and if you like my YouTube, then don't forget the subscribe to my channel. So again, thank you very much for listening.